Welcome to the introduction to the digestive system. This system is more than just eating and breaking down food. It's the foundation of how energy and every cell in our body receives the energy and nutrients it needs to survive. Think of it as a long coordinated assembly line that transforms a meal into fuel. We'll move through the anatomy and physiology of this system, exploring how each organ contributes to digestion, absorption, and waste elimination. The digestive system might seem very simple on the surface. We eat, we digest, we excrete. But it's surprisingly complex. Every bite of food you take sets off a chain reaction involving muscle contractions, enzyme activity, and intricate neural and hormonal regulation. What's amazing is that this process happens automatically without us even thinking about it. The system works continuously to maintain energy balance and homeostasis. As we go through this chapter, I want you to think about how digestion connects to other systems. Let's start with the big picture. The digestive system is made up of two main groups of organs: the digestive tract, also known as the alimentary canal, and the accessory organs. The tract itself is a continuous tube running from the mouth all the way into the anus, about 25 feet long in a living person. Accessory organs like the liver, pancreas, and salivary glands don't directly carry food, but they play key roles through their secretions and mechanical help. This system performs five main functions: ingestion, propulsion, digestion, both mechanical and chemical, then absorption and defecation. Here's the interesting fact. Food technically remains outside of your body until it crosses the intestinal lining. The lumen of the GI tract is considered external, which is why the body has to regulate this passes through all carefully. Now let's take a closer look at the tract itself. We'll start with the mouth. Move through the pharynx and the esophagus, then into the stomach. From there, food travels through the small intestines, divided into the duodenum, jejunum, and ileum, and finally through the large intestines, ending at the rectum and anus. Each section is specialized for certain tasks. For example, the stomach is designed for churning and protein breakdown, while the small intestines is built for nutrition absorption. Even though it's one continuous tube, every region has unique structural modifications that reflect its function. Supporting this whole process are the accessory organs. In the mouth, we have teeth, tongue, and the salivary glands, which prepare food mechanically and chemically before it's even reached in the stomach. In the abdomen, the liver, gallbladder, and pancreas take over. The liver produces bile to help digest fats, and the gallbladder stores the concentrates that bile. And the pancreas releases powerful digestive enzymes and bicarbonate to neutralize stomach acid. Together they form a team that ensures nutrients are fully broken down and ready for absorption. Now that we know the major organs, let's zoom into the microscopic level. Every section of the digestive tract, whether it's the esophagus or the small intestines, shares a common structural plan built in four concentric layers: the mucosa, submucosa, muscularis externa, and the serosa or adventitia. Each of these layers plays a role in digestion and protection. The mucosa is where absorption and secretion happens. The submucosa carries blood vessels and nerves as well as glands. And the muscularis externa handles movement, and the outermost layer, the serosa or adventitia, anchors and protects the tract. The mucosa is the layer in direct contact with food, so it's constantly working and constantly under stress. It has three sublayers: the epithelium, the lamina propria, and the muscularis mucosa. The type of epithelial tissue changes along the tract: stratified squamous in areas exposed to friction, like the mouth and esophagus, and simple columnar in regions that handle absorption, like the stomach and intestines. Goblet cells secrete mucus for protection, while enteroendocrine cells release hormones that help coordinate digestion. Beneath that, the lamina propria is rich in blood vessels and lymphatics, providing nourishment and immune defense, and the muscularis mucosa creates folds that increase surface area. Below the mucosa lies the submucosa, the supportive layer made up of areolar connective tissue packed with blood vessels, lymphatics, and glands. It's home to the submucosal plexus, which is part of the enteric nervous system that regulates local secretions of blood flow. Then comes the muscularis externa, which is the muscle engine of the GI tract. It usually has two layers: the inner circular layer and the outer longitudinal layer, that work together to produce peristalsis and segmentation. Between them is the myenteric plexus, which is another part of the enteric nervous system that controls motility. The outermost layer depends on where the organ sits. Inside of the abdominal cavity, most of the organs are covered in serosa, which is a smooth, slippery membrane that reduces friction as the organs move. It's part of the viscoperitoneum. Outside of the cavity, like the esophagus, the outer layer is called the adventitia, made up of dense connective tissue that anchors to the organs in place. Both layers provide protection, but while the serosa helps with mobility and lubrication, the adventitia focuses on stability and attachment. The digestive system has its own nervous network, the enteric nervous system, sometimes called the second brain. It has around 100 million neurons, almost as many as the spinal cord, and it can function independently from the central nervous system. The two main plexuses are the myenteric plexus, which controls muscle contractions, and the submucosal plexus, which controls secretions and blood flow. This system coordinates peristalsis, segmentation, and even reflexes like swallowing and vomiting. Even though the enteric nervous system works independently, it still communicates with the autonomic nervous system, the body's main control network. The parasympathetic division, often called rest and digest, boosts digestive activity. It increases motility and secretions mostly through the vagus nerve. On the other hand, the sympathetic divisions, our fight or flight response system, does the opposite. It slows digestion down, conserving energy for emergencies. Like any busy system, the digestive organs need a steady supply of blood, not just to feed themselves, but to carry out absorbed nutrients. The arteries that branch from the abdominal aorta deliver oxygen and nutrients to the organs, while veins collect nutrient-rich blood and send it through the hepatic portal vein to the liver. The liver processes, detoxifies, and stores nutrients before sending the blood into circulation. This routing through the liver is what keeps toxins from going straight into the heart and the brain. Lipids take a different route through this lymphatic system, entering tiny vessels called lacteals before joining the bloodstream. The peritoneum is a large double-layered serous membrane that lines the abdominal cavity and covers most digestive organs. It has two layers, the parietal peritoneum, which lines the body wall, and the visceral peritoneum, which covers the organs. Between them lies the peritoneal cavity, a space filled with lubricating fluid to prevent friction as organs slide and contract. Some organs, like the stomach and small intestines, are completely surrounded by peritoneum. We call this the interperitoneum. Others, like the pancreas and part of the colon, sit behind it. Those are retroperitoneal. Mesenteries are extensions from the visceral peritoneum that connect organs to the body wall and to each other. They stabilize organ position while still allowing movement, and they act as passageway for blood vessels, nerves, and lymphatics. They might recognize the greater omentum, the large apron-like fold of fat that drapes over the intestines. It cushions and insulates the abdominal organs. The lesser omentum connects the stomach to the liver, while the mesentery proper anchors to the small intestines. Finally, let's review what the digestive system actually does. It performs 7 essential processes: ingestion, propulsion, chemical and mechanical digestion, secretion, absorption, and defecation. Each step is precisely timed and coordinated between the organs. Ingestion brings food in, propulsion through peristalsis and segmentation moves it along. Mechanical digestion physically breaks down food, while chemical digestion uses enzymes to break it into molecules smaller enough to absorb. Secretion adds water, enzymes, and amino acids and bile to the mix. Absorption moves the nutrients into the bloodstream or lymph, and defecation eliminates what's left. Now let's talk about how food actually breaks down. Mechanical digestion is all about movement. It's the physical breakdown of food into smaller pieces. Think chewing, churning, and segmentation. This process increases surface area so enzymes can work more effectively. Chemical digestion, on the other hand, uses enzymes to dismantle food molecules into their building blocks. Proteins into amino acids, carbs into simple sugars, and fats into fatty acids. It starts right in the mouth with enzymes like amylase and continues in the stomach and small intestines. The GI tract doesn't just sit still, it's constantly in motion. Propulsion refers to the movement of food through the tract, mainly by peristalsis. Those wave-like contractions of smooth muscle that push food forward, even if you're hanging upside down. It starts in the esophagus and continues through the intestines. Segmentation, on the other hand, doesn't move food forward. Instead, it mixes the contents, bringing chyme in contact with the intestinal wall for better absorption. These motions are coordinated by the enteric nervous system, particularly the myenteric plexus, which controls movement contractions. Each organ in the digestive system contributes something unique to the process. The mouth handles ingestion and mechanical breakdown. The stomach churns and starts protein digestion. The small intestines complete digestion and absorb nutrients, and the large intestines reclaims water and compact wastes. The accessory organs like the liver and pancreas release secretions that support these steps. The digestive system is constantly adapting to what's happening inside. Neural control starts with sensory receptors in the GI wall that detect stretch, chemical composition, and osmolarity. These signals can trigger short reflexes, which are handled entirely within the enteric nervous system, or long reflexes that involve the brain and autonomic nerves. Short reflexes coordinate local activity like secretions and peristalsis, while long reflexes respond to external stimuli like smell or sight of food. Alongside nerves, hormones play a big role in coordinating digestion. Specialized enteroendocrine cells in the stomach and small intestine release hormones directly into the blood. Gastrin stimulates acid secretion in the stomach. Secretin promotes the pancreas to release bicarbonate and cholecystokinin, also known as CCK, which triggers the release of bile and pancreatic enzymes. Meanwhile, GIP, which helps slow gastric emptying into the intestines, have time to process the nutrients. These hormones make sure digestion happens in sync, each one switching on and off certain parts of the system to match the stage of digestion. Now let's move into the upper digestive tract, the oral cavity, oropharynx, and esophagus. These are the main entry points of the alimentary canal, and they're where food first meets the body's machinery. Here, mechanical digestion starts the chewing, chemical digestion begins within the saliva, and the bolus forms before swallowing. These regions also handle sensory functions like taste and texture, and protect the airway during swallowing. The oral cavity is the stage where ingestion begins. It's framed by the lips, cheek, palate, and tongue. The hard and soft palates form the roof, and the tongue forms the floor. This space is lined with non-keratinized stratified epithelium, tough enough to handle rough foods, but more soft enough for comfort. The oral cavity handles multiple functions like chewing, tasting, breathing, and speaking. The palatine tonsils near the back act as the part of the immune system, which protects the entrance to the pharynx. The tongue is far more than just a muscle that talks. It's a dynamic organ made up of skeletal muscle covered by a mucous membrane attached to the mandible, hyoid, and temporal bones. Its surface is dotted with papilla, which give texture and house taste buds. The tongue's intrinsic muscle changes its shape for speech and swallowing, while the extrinsic muscles move it into position for food during chewing. Its movement helps the food bolus and push it towards the back of the pharynx to begin swallowing. Next, the salivary glands. These are the body's first chemical factories in digestion. There are three major pairs, the parotid, submandibular, and sublingual glands, plus many smaller ones together in the mouth. Together they produce saliva, about 1 liter per day, mostly water but rich in amylase, mucus, and protective molecules like lysozymes and IgA. Saliva moistens food for easier swallowing, starts carbohydrate digestion, and helps control bacteria in the mouth. And of course, digestion wouldn't start without teeth. Humans have two sets, deciduous or baby teeth and then permanent teeth. Their shape matches their function. Incisors cut, canines tear, and molars grind food into smaller pieces. The visible part of the crown is coated in enamel and the hardest substance in the body. Beneath it lies the dentin and the pulp cavity filled with blood vessels and nerves. The roots are anchored to bone and the periodontal ligament and covered in cementum. Once food leaves the mouth, it enters the pharynx, the shared passageway for both air and food. The pharynx has three regions, the nasopharynx, oropharynx, and laryngopharynx. Only the lower two are involved in digestion. These are lined with non-keratinized stratified squamous epithelium to protect against abrasion as food passes through. During swallowing, the soft palate closes off the nasopharynx and the epiglottis folds down to block the airway, preventing food from entering the trachea. Muscles in the pharyngeal walls then contract to push the bolus towards the esophagus. The esophagus is essentially a muscular highway connecting the pharynx to the stomach. It's about 25 centimeters long and lies behind the trachea, passing through the diaphragm via the esophageal hiatus. It has two sphincters, the upper esophageal sphincter, which controls the entry of food into the lower esophageal sphincter, which prevents acid from following back to the stomach. The upper part contains skeletal muscle, giving us voluntary control when we start swallowing, while the lower section transitions to smooth muscle for automatic propulsion. The esophageal wall is designed for both protection and flexibility. The mucosa features a non-keratinized stratified squamous epithelium that resists friction from rough food. Beneath that, the lamina propria houses mucus-secreting glands to keep it lubricated. The submucosa supports this layer with blood vessels and more glands, while the muscularis externa transitions from skeletal to smooth muscle along its length. The outermost layer is an adventitia, which is a fibrous connective tissue that anchors the esophagus to the surrounding structures. Swallowing or deglutition is a beautifully coordinated event involving more than 20 muscles. It happens in 3 phases. First, the voluntary phase, when the tongue pushes the bolus into the oropharynx. Then the pharyngeal phase, which is involuntary. This is when the soft palate closes off the nasal passage and the epiglottis covers the airway. Finally, the esophageal phase uses peristaltic waves to move food down to the stomach. The swallowing centers in the medulla and pons coordinated this process, with several cranial nerves involved. Clinically, problems in any phase can cause dysphagia or difficulty swallowing. Conditions like GERD result from where the lower esophageal sphincter is not closing properly, allowing acid reflux. Achalasia is another disorder where the sphincter fails to relax, trapping food in the esophagus. The stomach is where digestion shifts into high gear. It sits just below the diaphragm and has 4 main regions: the cardia, fundus, body, and pylorus. Food enters through the cardia and mixes and churns in the body, and exits through the pylorus into the small intestines. The pyloric sphincter controls this exit carefully, letting only small amounts of chyme through at a time. The stomach shape is defined by its greater and lesser curvatures, and when it's empty, the inner linings folds into the rugae. It can expand dramatically from about 50 millimeters when empty to several liters when it's full. The stomach wall follows the same 4-layer plan as the rest of the GI tract, but adds a twist, literally. Its muscularis externa has 3 layers instead of 2, longitudinal, circular, and an extra oblique layer. This gives the stomach's incredible churning power to mix food and gastric juices. The mucosa contains deep gastric pits leading to gastric glands lined with secretory cells. When empty, the mucosa folds into visible rugae that disappear in the stomach fills. The submucosa provides elasticity and vascular support, and the outer serosa helps the stomach glide smoothly against other organs. Inside the gastric glands, we find 4 key cell types working together to create gastric juices. Parietal cells secrete hydrochloric acid, which helps break down food and kill pathogens, and intrinsic factor, which is essential for vitamin B12 absorption. Chief cells release pepsinogen, an active enzyme that becomes pepsin in acid, beginning protein digestion. Mucous neck cells produce a thin mucus to lubricate the stomach's contents, while endocrine cells, like G cells, release gastrin, a hormone which stimulates acid secretion and motility. The stomach performs both mechanical and chemical digestion. Its muscular contractions, called mixing waves, occurs about every 20 seconds, transforming food into chyme, which is a thick acid paste. Chemically, hydrochloric acid denatures proteins and activates enzymes like pepsin. Lingual lipase from the mouth begins digesting fats here, while carbohydrate digestion pauses because salivary amylase is inactivated by the acid. Only a small amount of chyme enters the small intestines at one time, about 3 millimeters per wave, allowing gradual digestion. Substances like alcohol, aspirin, and certain drugs can be absorbed directly through the stomach lining. The stomach's activity is controlled by three overlapping phases: the cephalic, gastric, and intestinal. The cephalic phase begins before food even arrives. Sight and smells triggers the vagus nerve stimulation, priming the stomach for action. The gastric phase starts when food stretches the stomach and raises its pH, activating gastrin and promoting acid and enzyme secretion. The intestinal phase kicks in when chyme enters the duodenum, initially stimulating digestion and then slowing down it through hormones like secretin and CCK. Neural and hormonal feedback keeps this process balanced. With all the acids and enzyme activity, the stomach needs serious protection. The mucosal barrier does exactly what it's made up of: thick alkaline mucus, tight junctions between epithelial cells, and rapid cell turnover. Entire surface cells are replaced every few days. If this barrier fails, it can lead to ulcers. The most common cause is caused by Helicobacter pylori infections and chronic NSAID use, both of which damage the mucosa. When ulcers erode deeply, they can cause bleeding or even perforations into the peritoneal cavity, leading to peritonitis. The small intestines is true workhorse of the digestive system. It's where nearly all digestion and absorption takes place. It stretches about 10 feet in a living person and nearly twice that in a cadaver due to the loss of muscle tone. Despite its name, the long and narrow, about 2.5 centimeters in diameter. When chyme enters the stomach through the pyloric sphincter, it's met with bile from the liver and pancreatic enzyme that completes the digestive process. Its lining provides an enormous surface area, about 200 square meters, roughly the size of a tennis court. The small intestine has three major regions: the duodenum, the jejunum, and the ileum. The duodenum is the shortest section, only about 10 inches long, and receives bile and pancreatic secretions through the major duodenal papilla. It also retroperitoneal, meaning it's anchored behind the peritoneum. The jejunum follows. It's a thick-walled, highly vascularized, and the main site of nutrient absorption. Finally, the ileum, which is the longest segment, connects to the large intestine at the ileocecal valve and contains Peyer's patches, which are clusters of immune tissues that protects against bacteria. Together, these regions ensures that the time chyme leaves, most nutrients have already been absorbed. The small intestine's histology is a masterpiece of efficiency. It has the standard 4 layers, but features special modifications to maximize absorption. Circular folds, or plicae circulae, which are the spirals the chyme slowly, increasing time for digestion. On the mucosal surface, villi rise like tiny fingers, each containing blood capillaries and lymphatic vessels called lacteals, and even smaller projections called macrovilli form a brush border on each epithelial cell. These three features together increase the surface area for over 600 times. The result is an incredibly efficient interface of absorbing nearly every nutrient the body needs. Between the villi lie intestinal glands called crypts of Leberkuhn, which secrete intestinal juice rich in enzymes and mucus. In the duodenum, Brunner's glands in the submucosa produce alkaline mucus to neutralize stomach acid. Goblet cells scattered throughout the epithelium secrete mucus, keeping the surface moist and lubricated. Enteroendocrine cells release hormones such as secretin and CCK to coordinate digestion with the pancreas and the gallbladder. The ileum's Peyer's patches act as immune sentinels, preventing harmful bacteria from invading the bloodstream. Food movement through the small intestines in this rhythm of contractions and relaxation. Segmentation mixes the chyme with digestive juices, enhancing contact with the mucosa for absorption. These contractions are most frequent in the duodenum and slow down in the ileum. Between meals, migrating motility complex sweep any leftover debris towards the large intestines. Chemically, pancreatic enzymes and brush border enzymes break carbohydrates, proteins, and fats into absorbable units. Bile from the liver emulsifies fats, increasing surface area for lipase to act. Almost all absorption takes place in the small intestines. Nutrients move through the epithelial cells into either the blood capillaries or lacteals. Carbohydrates and proteins enter the bloodstream as simple sugars and amino acids, while fats travel through the lymphatic system as chylomicrons. Vitamins, electrolytes, and water also cross the mucosa here. The small intestines function as a highly coordinated unit, balancing digestion, absorption, and immune protection. When the function is compromised, it's called celiac disease or Crohn's disease. Nutrient deficiencies can occur even if a person eats well. Now we move on to the large intestine, the final stage of digestion. Although only about 5 feet long, it plays a crucial role in reclaiming water and electrolytes, synthesizing vitamins through bacterial actions, and compacting waste into feces. Unlike the small intestines, its job isn't digestion but conservation. It begins in the ileocecal valve and ends in the anus. The large intestine also houses trillions of beneficial bacteria that perform fermentation and regulate immune function. Anatomically, the large intestine forms a frame around the small intestines and has 4 main regions: the cecum, the colon, the rectum, and the anal canal. The cecum is the entry point receiving chyme through the ileocecal valve. The colon itself has 4 sections: the ascending, transverse, descending, and sigmoid. It's wider than the small intestines but shorter. The rectum stores feces, and the anal canal is where defecation occurs. The ileocecal valve prevents backflow, ensuring one-way traffic. The cecum is the pouch-like structure just below the ileocecal valve. It continues the absorption of water and electrolytes that begin in the small intestines. Attached to it is the appendix, a small, narrow, worm-like tube containing lymphoid tissue. Though often considered vestigial, recent research suggests it may serve as a reservoir for beneficial gut bacteria and plays a minor immune role. When the appendix becomes inflamed or obstructed, it leads to appendicitis, which is a painful and potentially dangerous condition if it ruptures. The colon makes up the majority of the large intestine and follows an almost square path around the abdominal cavity. It starts on the right with the ascending colon, bends to the hepatic flexure and crosses to the transverse colon, then turns again in the splenic flexure to the descend on the left before it curves onto the sigmoid colon. Most of the colon is retroperitoneal, lying against the back body wall. Its main function is to absorb water and electrolytes, compact fecal material, and move it forward through slow, steady peristalsis and hydral contractions. At the end of the line, we reach the rectum and anal canal, the final stretch of the digestive tract. The rectum serves as a temporary storage site for feces, equipped with 3 internal folds that separate gas from solid materials. And yes, that's why we can usually tell the difference. The anal canal is about 2 inches long and surrounded by 2 sphincters, the internal and external sphincters. The internal sphincter made up of smooth muscle which operates involuntarily, and the external sphincter made up of skeletal muscle which we control voluntarily. Together, they regulate defecation. Histologically, the large intestine differs from the small intestines in that they lack villi and circular folds. Absorption here is limited mainly to water and electrolytes. The mucosa is lined with simple columnar epithelium, rich in goblet cells that secrete mucus to keep fecal material moving smoothly. Between the cells are deep intestinal folds packed with absorptive and mucus-secreting cells. The submucosa holds blood vessels and lymphatic tissues, while the muscularis layer features the tinea coli, 3 longitudinal muscle bands responsible for haustral pouches. The colon has 3 distinct surface features: the tinea coli, which run along the length, maintaining muscle tone and assisting with mass movements. When the bands contract, they form haustra, which are sac-like pouches that slow down, transmit, and allow more water absorption. Attached to the outer surface are small, fat-filled sacs called epiglottic appendages, which function is not nearly entirely clear, but may relate to cushioning or immune response. In contrast, the rectum and anal canal lack tinea coli, instead having thick continuous muscle layers that handle the force of defecation. The large intestine is home to a vast and diverse bacterial community, over 700 species strong. These microbes ferment undigested carbohydrates, produce gases like hydrogen and methane in the process. They also synthesize essential vitamins such as vitamins K, biotin, and pantothenic acid. Beyond digestion, they help train the immune system and protect against pathogens. In return, the body provides them with a nutrient-rich environment. This symbiotic relation is crucial for health, Disruptions such as from antibiotics or poor diet can lead to issues like bloating or even inflammatory bowel disease. Unlike the constant motion of the small intestines, the large intestine moves more slowly. House contractions occurs every 30 minutes or so, mixing the contents and promoting water absorption. Peristaltic waves continue this process, but the most dramatic movements are the mass movements, strong wave-like contractions that push fecal matter towards the rectum. They are more often triggered after meals by the gastrocolic reflex, which explains why some people feel the urge to go shortly after eating. This system keeps the flow steady but deliberate, allowing time for maximum absorption. By the time materials reach the large intestine, about 90% of the water has already been absorbed in the small intestines. The large intestine reclaims most of what remains, transforming liquid chyme into semi-solid feces. Feces contains water, undigested food, and bacteria as well as waste products. The defecation reflex begins with the stretch receptors in the rectal wall detect fullness. Parasympathetic signals contract the rectum and relax the internal sphincters, while voluntary control of the external sphincters determines the timing of release. Now let's turn our attention to the accessory digestive organs, the liver, gallbladder, and pancreas. Though food never passes through them, they play a vital supporting role. The liver produces bile to emulsify fats, the gallbladder stores and concentrates that bile, and the pancreas secretes digestive enzymes and bicarbonate-rich fluid into the duodenum. These organs ensure that fats, proteins, and carbohydrates are fully broken down for absorption. The liver is the largest internal organ, weighing about 3 pounds and sitting in the right upper quadrant beneath the diaphragm. It has 4 lobes, the right, left, caudate, and quadrate, and it's held in place by the ligaments like the falciform and the lesser omentum. The liver has a unique dual blood supply. Oxygenated blood comes from the hepatic artery, and nutrient-rich blood from the digestive tract arrives via the hepatic portal vein. These vessels meet at the porta hepatis, the liver's central hub, where the common hepatic duct also acts as carrying bile. Structurally and functionally, the liver is a metabolic powerhouse. Microscopically, the liver is arranged into hexagonal units called lobules. Each lobule contains rows of hepatocytes, the main liver cells, radiating around a central vein. At each corner of the lobule sits a portal triad, a branch from the hepatic artery, a branch from the hepatic portal vein, and a bile duct. Blood from both sources flows through the sinusoids, leaky capillaries, where it's filtered and processed by the hepatocytes before draining into the central vein. Meanwhile, bile flows out the opposite direction through tiny channels called bile canaliculi. The liver produces about 1 liter of bile each day, which is a fluid critical for fat digestion. Bile contains bile salts, pigments like bilirubin, cholesterol, phospholipids, and electrolytes. Its main function is to emulsify fats, break down large droplets into smaller ones to increase surface area for enzymes like lipase. Bile flows from hepatocytes into canaliculi, then through the bile ductules to the right and left hepatic ducts, which merge into the common hepatic duct. From there, it moves either directly to the duodenum or stored in the gallbladder via the cystic duct. Bile released is triggered from the hormone CCK when fatty chyme enters the small intestine. The gallbladder is a small pear-shaped sac tucked beneath the right lobe of the liver. Though small, its job is mighty. It stores and concentrates bile produced by the liver between meals. When we eat fatty foods, the hormone CCK signals the gallbladder to contract, releasing bile through the cystic duct into the common bile duct, which opens into the duodenum. Histologically, the gallbladder has folds like rugae that allow expansion and smooth muscle layers that squeeze bile out when needed. Without bile, fat digestion would be incomplete, and fat-soluble vitamins like A, D, E, and K couldn't be absorbed properly. Clinically, gallstones can form if bile components crystallize, leading to painful blockages or inflammation. The pancreas sits retroperitoneally, nestled behind the stomach and connected to the duodenum. It has 3 parts, the head, body, and tail. Functionally, it's a dual-purpose organ, most exocrine but partly endocrine. The exocrine portion is composed of acinar cells that produce digestive enzymes, and these secretions travel through the duct system that merge with the bile duct at the hepatopancreatic ampulla. The pancreas produces about a liter of pancreatic juice per day containing water, bicarbonate, and enzymes like amylase, lipase, and proteases. The bicarbonate neutralizing acidic chyme form from the stomach, creating an optimal pH for enzymatic activity in the small intestines. Pancreatic juice is a finely tuned chemical cocktail. Its high bicarbonate content buffers stomach acid while enzymes take apart micronutrients. Amylase breaks down starch into sugars, lipase digests fats, and nucleases break down nucleic acids. Proteases like trypsinogen and chymotrypsinogen are released into inactive forms and activated only if the duodenum to prevent the pancreas from digesting itself. Enteropeptidase, an enzyme in the small intestine, starts this activation chain. Its remarkable safeguard: when the processes malfunction, as in pancreatitis, some enzymes can begin digesting pancreatic tissues. The pancreas also has an endocrine side made up of clusters called islet of Langerhans. These contain several types of hormone-secreting cells: alpha cells that release glucagon, beta cells that produce insulin, delta cells that secrete somatostatin, and PP cells which make pancreatic polypeptides. Together, these hormones regulate blood glucose levels and coordinate energy use. Insulin lowers blood glucose by promoting uptake in the cells, while glucagon raises it by stimulating glycogen breakdown. When this balance fails, as in diabetes mellitus, the body can't properly regulate blood glucose. The pancreas can also be affected by inflammation, enzyme blockage, or genetic conditions like cystic fibrosis, all of which disrupt digestion. This organ's dual role, digestive and endocrine, makes it one of the most vital in the entire body.